Welcome to today's lesson on cellular respiration. Today we're going to talk about two of the steps. We're going to talk about the Krebs step and the Krebs cycle. But before we go into that, we're going to briefly go over some words that we should know. Oxidation is the loss of an electron. So the opposite of that is reduction, the gain of an electron. And then lastly, an isomer is the same formula, just different arrangement. So they have the same number of each atom, just rearranged differently. And so you'll hear me talk about those later on in this presentation. So if we recall, glycolysis is an anaerobic step, and so it does not require oxygen. And it happens in the cytoplasm of the cell, and the 6-carbon glucose molecule ends as 2 3-carbon pyruvate molecules. From there forward, everything is aerobic, meaning it requires oxygen. So first off, we have the Krebs step, where the 2 3-carbon pyruvate molecules are transported into the mitochondria through the intermembrane space and through a transport protein into the matrix. And therefore, from there on, it is oxidized, so it becomes from the 3-carbon pyruvate to a 2-carbon acetyl group. The 1 carbon that's lost is picked up as CO2 and transported out of the mitochondria into the cytoplasm. And also, NAD becomes reduced and picks up that hydrogen and becomes NADH. Along with that, coenzyme A comes along and binds with the 2 acetyl groups, 1 for each pyruvate molecule. And so we end up with acetyl-CoA in the Krebs step. Now, into the citric acid cycle, which both happen in the matrix. We are just taking this from the matrix and making it bigger outside, but just as I'm going on, just remember it's in the matrix. The acetyl groups are carried into the citric acid cycle, where the CoA is released and the acetyl group bonds with a 4-carbon molecule called oxybatate and water, and it becomes a 6-carbon citrate molecule. From there, it is rearranged into an isomer. Remember, as I recalled earlier, the definition is same formula, just different arrangement. So it becomes isocitrate, which is still the 6-carbon molecule. And then from there, isocitrate is oxidized, we lose a carbon, and it forms CO2 and is released. And NAD reduces, picks up that electron as a hydrogen molecule and forms NADH. So we lost that carbon, so it becomes a 5-carbon molecule. And then once again, it's oxidized, so we lose another carbon as CO2. NAD picks up an electron as hydrogen, forms NADH, and we now have a 4-carbon succinyl CoA group. Remember, I said CoA, so that CoA is now released, and the energy that is gained from releasing the CoA turns GDP into GTP, which, due to substrate level phosphorylation, turns ADP into ATP, which is the only ATP made in the citric acid cycle, also known as the Krebs cycle. From there, we went from succinyl CoA to succinate, 4-carbon molecule still, and we become oxidized once again, losing 2 electrons and 2 protons. But this time, instead of NAD coming in, FAD comes in and now becomes FADH2. So we go from succinate to fumarate, and then we add water, turning fumarate into malate. Well, malate becomes oxidized, and NAD once again becomes NADH, and malate turns into oxaloacetate. And as I said earlier, that reacts with the acetyl molecules from the Krebs step. So all it has to do is react with another set of acetyl coming in from the Krebs step to re-enter the cycle. And that's brief description of the Krebs step and the Krebs cycle.